Hey guys, welcome back to Neuronify. This is day 3 of our FMT-UT series and the topic is identification. So in this video, we'll be covering all the important questions regarding identification for university exams. Our chapter is identification and our very first question is, describe the steps of forensic DNA fingerprint. DNA fingerprinting is also called as DNA finger profiling. The DNA molecules are present in chromosomes about 99% and mitochondria 1%. They are very long. DNA in a human cell is about 1 meter chain and is composed of about 500 crore subunits called as nucleotides, each of which is composed of deoxyribose sugar and phosphate molecules that are twisted into double helix and are bound together by links of heterocyclic base of adenine, thymine, guanine and cytosine. Adenine of one chain pairs with thymine of the other and cytosine of one with guanine of other. The DNA chains are thus in pairs of two strands of polynucleotides, which is double helix. So this is all about the introduction regarding the structure of DNA molecules. During formation of spermatozoa and ova, the DNA strands separate and thus due to the fertilization in the offspring, there is strand contributed by each parent. Matching of nucleotide sequence of strands of one cell with those of another implies that the two are from same human beings. Since the DNA print for an individual is unique as fingerprints, the method is called as DNA fingerprinting. So as we have got unique fingerprints, we have also got unique DNAs. So why there is concept of DNA fingerprinting. However, uniovular twins have same sequence. When a small quantity of DNA chain is available from a cell, then by PCR method, its quantity can be increased. What is the source? Nucleated cells from blood like WBCs, semen, vaginal epithelium, tooth bulb, bone marrow, hair roots, muscles, skin and mucous membrane, etc. Material should be dry and if liquid, it should be frozen. What are the medico-legal aspects of DNA fingerprinting? Identification of accused from DNA fingerprinting of blood and hair found at scene of crime. Number 2. Identification of accused of rape from DNA fingerprinting of semen or spermatozoa. Identification of multilated bodies. DNA is much more stable and therefore even old stains can be typed. Identification of old dead body remains by comparing with DNA of his parents or wife and child or DNA specimen from toothbrush, biopsy slides or licked envelopes or stamps. Cases of disputed paternity by studying the DNA pattern in child, mother and disputed father. Case of disputed maternity, cases of babies mixed in hospital. Missing persons can be identified by matching their DNA with their parents. This study is yet to be accepted by courts as evidence. It is costly, lengthy process, requires 10 weeks and requires expert opinion only. So this was about the DNA fingerprinting. First is biological material we have to take, then we have to isolate the nuclei, then isolate and purify DNA, digest DNA with restriction enzymes, separate DNA fragments by gel electrophoresis, then sudden transfer of DNA into nylon membrane, then hybridization to label DNA probes, then wash membrane, autoradiography of DNA parents and visual and computer analysis of DNA profiles. So this is the diagram of DNA fingerprinting. In DNA fingerprinting, remember you just have to write the other name, which is DNA finger profiling also, and just describe what is the structure of DNA molecule in brief and describe about the bonds between the DNA strands, adenine, thymine and guanine and cytosine. Describe the source also, and main thing is medical legal aspect. What is the medical legal aspect? Identification of accused from DNA fingerprinting of blood and hair found at scene of crime. Identification of accused of rape also. Multilated bodies, paternity issues, maternity issues, cases of babies mixed in hospital, missing persons, and remember that it is very costly and it is lengthy process. Moving ahead, what is Gustafson's method of dental examination, age determination. So what is Gustafson's method? This method considers the changes in the longitudinal section of the tooth with advancing age. The changes are, they just want to say that this method considers certain changes in the longitudinal section of the tooth with advancing age, with increase in age. The changes are: number 1, attrition by mastication, that is tooth destruction because of chewing seen on masticating surfaces. A0 nil, A1 attrition up to enamel, A2 attrition up to dentine, and A3 pulp exposed. So these are the stages, levels of the attrition by mastication. What is mastication? Chewing. Chewing is called as mastication. So attrition, which means destruction caused due to chewing. There are 4 levels: A0, A1, A2 and A3. So in A0 we have nil, that is there is no damage. In A1 attrition up to enamel. For that reason, first understand the diagram of tooth. Number 1, this is at the bottom left side, root is present. Then above root we have neck and crown. And enamel is the outermost part of the tooth. Then below it is dentine, then dental pulp. We have gum also, alveolar process, periodontal membrane and cementum. So what is A0? No damage. What is A1? Attrition to enamel. Dentine A2 and pulp exposed A3. What is periodontosis? Periodontosis, that is regression of gums, therefore loosening of teeth and therefore root becoming visible. So periodontosis is nothing but decrease in the size of gums. That is nothing but regression of gums before loosening of teeth. Therefore loosening of, obviously if gums are going to become loose, then if gums are going to decrease in size, then there would be loosening of tooth. That is why they said in periodontosis there is decrease in the size of gums, therefore loosening of teeth and therefore root becoming visible. Root is present at the bottommost site. What is P0? No root exposed. P1 1/3 root exposed. P2 1/3 to 2/3 root exposed, and P3 more than 2/3 root exposed. So talking about Gustafson's method, just remember in single language, anything which describes about any method which describes about the longitudinal section of tooth, and may it be your enamel, may it be because of chewing or because of a decrease in size of gums. Third point is secondary dentine deposition in the pulp cavity. SO, S1, S2, S3. So every point has got 4 levels, remember, starting from 0 to 3. So talking about our third point, secondary dentine deposition in the pulp cavity. So dentine deposition is observed in the pulp cavity downwards. Pulp cavity is present below the dentine. So S0 is nil, S1 is on upper part in lower jaw and vice versa. And S2 is half of pulp is occupied and S3 is almost whole of the pulp occupied. Number 4, cementum apposition, that is thickening of cementum occurring on root surface. So cementum becomes thick. CO, C1, C2, C3 normal, slightly more than normal, thick layer present, heavy layer present. What is root resorption? R0, R1, R2, R3. R0 nil, R1 at some spots, R2 over larger areas, R3 more extensive. Root transparency, T0, T1, T2, T3. T0 nothing, T1 just noticeable, T2 up to 1/3 of apical area, and T3 up to 2/3 of apical area. The total score is added and is represented as X. Age is equal to 11.43 plus 4.56 into X. So this is the formula. You just have to add the total score and put the value in place of X. Alternatively, age can be known from plotting the score X on regression line. Only changes A and B can be seen on tooth surface, and for C to F, the tooth has to be removed from socket and its LS, that is longitudinal section, should be studied. Therefore, the study has only limited value in living. Yes, so what they want to say is A and B points are practically possible in living human being also. But for C to F points, we just have to remove the tooth and then we have to observe the changes. So what are the 6 points in this question? Very first thing, you just have to define what is Gustafson's method. It is nothing but observing and studying the longitudinal section of teeth or tooth. Then we have got 6 points in it. Number 1 is we just have to take care of mastication, which is chewing process. Number 2, regression of gums. Number 3, regarding dentine deposition, just understand the keywords. Number 4, thickening of cementum. Number 5, resorption of root. And number 6, root transparency. So these are the 6 points, and under every point we have got 4 levels from 0 to 3. Remember this. And there is one formula, age is equal to 11.43 plus 4.56 into x. What is x? Total score addition, total addition of all the 6 points. Fine. Moving ahead to the next question, what is anthropometry system of identification? Anthropometry, Bertillon's system of identification. Other name is Bertillon's or Bertillon's system of identification. Anthropos, human, metron, measurement. What is the principle? Is that body measurements do not change after 21 years. And what is the principle of this method or this system says that body measurements do not change after 21 years. And what is the procedure? The study includes complexion, facial features and photographs. Number 2, body peculiarities like scars, moles and deformities. Number 3, body measurements. Following 11 measurements are taken. Basically, in body measurements, 11 measurements are taken. Standing height, sitting height, distance between tips of middle fingers and outstretched upper limbs, length and breadth of head, width of face, length and width of right ear, length of left forearm, length of left hand, length of left middle finger, length of left little finger, length of left foot. So these are the 11 measurements taken in anthropometry system of identification. Anthropometry, anthropos is human and metron is measurement. What is the principle? It says body measurements do not change after 21 years. And the study includes complexion, facial features, photographs, body peculiarities like if the person is having any kind of moles, scars, deformities, then body measurements, and in body measurements we have 11 points. Standing height, sitting height, okay, distance between the tips of middle fingers and outstretched upper limbs, length and breadth in the head, and everything about the left hand. What is that? Length of left arm, forearm, left middle finger, little finger, left foot, and even the length and width of right ear. Fine. Questions? But first question is very important regarding forensic DNA fingerprinting. You just have to memorize the steps and you should draw a diagram in exam also. There are in total 14 steps. Biological material we have to collect, then we have to isolate nuclei, isolate and purify DNA, then digest DNA with restriction enzymes, separate DNA fragments by gel electrophoresis, sudden transfer of DNA into nylon membrane, hybridization to labeled DNA probes, then we have to wash membrane, and we have to do autoradiography of DNA patterns, and then visual and computer analysis of DNA profiles. So these are the steps in DNA fingerprinting. Very important question. Do not forget to write the introduction of DNA structure. And in this question, medical legal aspects are very important. In total, we are having 11 medical legal aspects, out of which main focus is given on the identification of accused, and also in cases of disparity or disputed maternity and paternity. So these were the questions, just 3 questions, and yes, we have completed identification chapter. Next chapter would be forensic thanatology. See you in the next video. Until then, keep learning, keep growing.